Certain species of sharks in Greenland can live over 500 years. So why can't we? Or maybe a better question is, can we? The answer today is no, but we have extended average lifespans quite a bit over the last 100 years. It is pretty clear that every species does have a genetic limit to lifespan. In humans, that might be around 120 if you look around at the supercentenarians out there. But in order for us to go from that number, 100, 120, whatever that is, to 500, that's going to require cracking the code of why we age and fix the flaw in the system, so to speak. But that's where things get really interesting. Even 20 years ago, the idea of increasing human lifespan by magnitudes rather than just a few years really was like pure science fiction. But now, there are many labs at major universities across the world who are working diligently on this problem with extraordinary results. And in my opinion, it's not a question of whether we will get there, but rather when. One of these scientists dedicated to solving the aging puzzle is Dr. Joel Rothman at the University of California, Santa Barbara, where he heads up the Center for Aging and Longevity Studies, and he's my guest this week on Sapio with Buck Joffrey. Now, Professor Joel Rothman is a distinguished professor of molecular, cellular, and developmental biology, the Wilcox Family Chair in Biotechnology, and the founding director of the Center for Aging and Longevity Studies at the University of California, Santa Barbara. Now, for 3 decades, his research group has explored embryonic development, programmed cell suicide, tissue reprogramming, and aging. He's been supported by many research grants from the National Institutes on Aging, the National Cancer Institute, the National Institutes of Child Health and Development, and other agencies. He's an elected fellow of the American Association for the Advancement of Science. With that, I give you Dr. Joel Rothman.
In pursuit of health and wisdom, Sapio with Buck Joffrey.
Joel, thanks for joining me.
Pleasure.
So tell me a little bit about yourself. What got you interested in the science of aging in the first place?
Well, probably the first discoveries that really transformed it from a rather fuzzy field, to be honest, where the science was not well defined, into an understanding that aging can be studied just like other biological processes that I've been trained in studying in molecular biology, cell biology, and so forth. That is to say that we can understand how proteins and pathways of proteins act together can act to actually program how rapidly animals age. That started with some really seminal studies in one of my favorite model organisms that I work on a lot, C. elegans, a very celebrated model organism that's won 6 Nobel Prizes and been celebrated for many other reasons. There are thousands of people working on them in the world, and the key discovery was made really initially in the 70s, but I became very aware of it in the 90s, particularly when Cynthia Kenyon's lab discovered that a single mutation could double the lifespan of the animals, and in so doing also extend the health span, the period of youthfulness in these animals. And from that, then working out a pathway, a molecular pathway responsible for that. And as soon as I saw that sort of pathway, I thought, okay, this looks like an area that we can really sink our teeth into.
Before that, I guess there was this sort of notion in general with scientists that there wasn't genes that you could alter that would change fundamentally lifespan or health span.
Yeah, I don't know if people would have said that per se, but still the feeling about aging was kind of a vague thing. It maybe reflected the second law of thermodynamics, for example, that is that things fall apart, that it couldn't be studied in the same way that, for example, cell division could be studied, or even cancer progression, you couldn't tackle it in that way. And I think those findings really, for me, nailed it, and I think for a lot of the community nailed the idea that, we can study aging and longevity the same way we can study all sorts of other cellular processes.
So, from the clinical side, coming at this as a physician, physicians ultimately operate under the premise that ultimately there's, you can do things to change longevity. There's low-hanging fruit. You can improve your blood pressure, you make your cholesterol appropriate, try to control your blood sugar, but ultimately that there is a hardwiring
Right.
of DNA that really ultimately says, okay, you've done a good job, but at some point here, whether that's 85 or 100 or maybe even 110, that's it.
Yep.
Do you agree with that?
Yes, I do, without changing our genome.
Right.
Without the mutations, for example, of the type I'm talking about. So what I mean by that is the things you're talking about, I strongly support and try to practice as much as I can. I would like to live longer. I love life.
Sure.
I want to stay healthy longer. But in the long view, those are sort of tweaks compared to what we can hope to achieve, which is greater than tweaks. We cannot achieve that without intervening in some way in our genetic code, either by changing gene expression in our bodies that is otherwise programmed by our own DNA, or by intervening in the products of our genes, that is to say through drugs, through pharmaceutical intervention. So the questions that drive us as basic scientists are really, how can we adapt the genome or the expression of the genome in a way that results in substantially bigger leaps, particularly in terms of extending health span. I mean, that's really what drives us most.
That's what's exciting to me about this space is, again, as a former clinician, and if you look at sort of human history in the last century or so, we have been able to live longer, right? Mm-hmm. And there are increments, in some cases, decades. But as you said, they tend to be tweaks. Whereas what you're talking about, and I think what basic science is ultimately addressing is, can we potentially increase lifespan and health span in magnitudes, right? And I guess the question for me is, if you look across the different organisms out there, you have such a huge varying lifespan, right?
Right.
Whether it's sharks, you could talk to this a little bit more than I can, but talk about that variability and what you think some of the things that go into that variability amongst different species is. Right,
so that's what convinced me as a geneticist that we can really make headway. If you just look at the natural world, the shortest living animals live weeks. In fact, C. elegans, the little creature we work on in the lab, lives about 3 weeks. The longest lived animals, there are claims that glass sponges live 10,000 years. Those are animals as well. But even in the vertebrate world, there's a huge variation. The shortest lived vertebrates, killifish, for example, live months, 4 to 6 months. The longest lived vertebrate that I'm aware of is the Greenland shark, which I think you alluded to. 500 years it's been estimated, 500, half a millennium. So there are Greenland sharks on the planet today that were probably alive when Magellan was sailing around the planet. Pretty mind-blowing. And even in the mammalian world, there are animals, bowhead whales, that live 220 years or so. And those differences ultimately have to be driven by genetics. It is the genomes of these animals that are driving these differences, because that's what makes these animals different. The thing that makes one species different from another in all of its traits is the genome. And so this radical difference in lifespan that we observe from 3 weeks up to half a millennium or even 10,000 years ultimately boils down to our genomes. We are humans, we are stuck with our human genomes, but we are also inventive and we can work with those genomes, and we have a lot of headspace there. We know that a mammal can live to 220 years. We know a vertebrate can live to 500 years. We know that an animal can live perhaps millennia. And so that tells me that we have the potential, even as a human species that lives a long time, but not as long as the longest mammal, for example, to not tweak, but really dramatically change the progress of the aging clock.
So there's a somewhat controversial figure, Aubrey de Grey, who I think you spent some time in a lab at the same time, some time ago, that he has a theory essentially. I don't know if it's a theory or just sort of this conceptual framework of this period in time where we may have what he calls longevity escape velocity.
Right.
Longevity escape velocity, essentially the concept there is, okay, you come into a period of time and you're 60 years old and you're very healthy, and we may start to get to a point where that 60-year-old person has this, this life expectancy of right now, another 78 to 80 years old, something like that. But those things that guy would die from would be solved, right? The problem, that issue would be solved before that 78th birthday.
Right.
Therefore, giving him or her another 10, 15 years to run into the next problem that would kill them.
Right.
And technology would constantly be ahead of the problem.
Yep. And you can keep leapfrogging ahead.
Right.
And ultimately, perhaps achieve immortality.
Yeah. What do you think of that?
So I think in concept, it's consistent with what we know about biology. And I'll say more about that in a second. And I'll just mention, I met Aubrey when he was a youngster, and I was a youngster just in passing. And I didn't study with him or anything like that, but I did meet him when he was in his 20s. So we've gone our own ways and grown a lot since and grown up a lot.
Yeah.
But I think that idea is biologically tenable, and the reason I say that is that, again, in the natural world and even in humans, we know that immortality is achievable. So what do I mean by that? Our gametes are produced from a lineage of cells that go back to the beginning of time. There is a part of us, we have immortality within us, and that is our germline, our gametes that go on to the next generation. And that there is a renewal process at every generation when a new offspring is conceived. There's a renewal. A baby is not, no matter how old the parent is not born as showing any signs of aging. They're brand new. And they came from our cells that were held within us. So we know biologically immortality is attainable from that standpoint. We also know from the modern advent of cloning procedures that it's also that the same is true. That is, if you clone let's say a sheep cell from an old sheep and you turn it into a new baby sheep, it's going to be a baby sheep. There is a refreshing, there is an immortality that can be invoked in biological systems. So there's nothing biologically that says immortality is unachievable, certainly at the cellular level. Now, the problem is the organismal level, and I think the one wrinkle in this notion, first of all, how long will it take? We don't know. We don't know how long it would be before we can keep leapfrogging ahead. But if you could extend your life 100 years, there's a lot of discovery that can be made in that time. So I don't think it's a ridiculous idea. The one question for me, there's one organ that concerns me, and that is our nervous system, our brain. We are, as individuals, so much a result of the structure of our brains, complex structure, that is not encoded. The ability to make a brain and make the wiring is encoded in the genome. But who we are as individuals, our experiences, our training, our learning, our individuality as personality, that's not encoded in the genome. That's the result of all the years that we've lived. Can we freshen up those cells, or can we bring in new young cells to replace those and not lose information content? And that's the one place that I think we might run into troubles in terms of this idea of achieving immortality by continuing to leapfrog ahead.
Sure.
And I don't think we have answers to that.
Yeah. So let's kind of dive into some of the mechanisms here, because I think I think this area, I think, of longevity, I think for the most part, I don't know if you agree with this, but it's all of a sudden hit popular culture, right? Like it's a buzzword, something that wasn't around 10 years ago. People weren't really talking about longevity as much, and this potential for living, magnitudes longer. And I think one of the people responsible for that was probably David Sinclair with his book Lifespan, which was a big hit, number one
certainly
seller.
Had a big impact. Yep.
And David obviously has done some really good work in this space. He has a uniform theory on aging. Why don't you talk about that? I think that's probably a good place to sort of start diving deeper into the science.
So I'll try to encapsulate this accurately, but he started, I've been aware of David's work from when he was a postdoc, in fact, in Lenny Guarenti's lab at MIT, and I was a professor at that point at the University of Wisconsin, and had followed his work from his postdoc years. He discovered in yeast that yeast lifespan could be extended by tweaking what turns out to be the epigenome. And I probably, for your audience, should define that. So I'll define that in a second. First of all, it's kind of weird even to think about yeast as aging, because yeast is a single cell organism that divides and buds off, and it just keeps dividing. And you think, well, how can this tell us anything about aging? But it actually has revealed some things about aging. The way they age is that when yeast cells divide, there's a bigger cell and a smaller cell, the mother cell and the daughter cell. The daughter cell is like the youngster. The mother cell can continue to divide a number of times, but they can't do that forever. So that's aging, how many times the mother cell can divide. And he found by tweaking, or by actually mutating a gene that's involved in controlling the epigenome in the end, that he could change the lifespan of those mother cells. So the epigenome, what does that mean? The genome is a description of all of the bits of information in our DNA. The 4 nucleotides, A, C, G, and T, the bits of information when we talk about the sequence of a genome, that's what we're learning about, the bits of information that are strung together in DNA. The epigenome very much affects how the genome acts, but it doesn't change that sequence. It doesn't change what you can write down on a piece of paper for all the bits of information. The epigenome, part of it is changes to DNA, but not in ways that change how it codes. They change how it's expressed. Another part of the epigenome is what's called the chromatin, which is the DNA and proteins that are all glommed together that control how well a gene is expressed. So what does it mean to say a gene is expressed? We have 20-some thousand genes, 26,000 or something like that genes in our genome. And it depends on how you divide that up. You can get each gene, many genes can be broken down into other pieces, and so you can consider them more genes, but we'll call it 26,000. Not all of those are expressed in any one of our cells. Some of those are on in liver cells to make a liver. Some of those are on in heart cells to make a heart, and so forth. And on means that they're actually producing the products that they code for. The genome is just a code. You have to read that code and turn it into something useful. That something useful is turned on only for certain genes in certain tissue types. The epigenome is instrumental in controlling which genes are turned on. Yeah. And that not only determines which genes are turned on or turned off in different cell types, like a liver cell or a heart cell or skin cell and so forth, but they're turned on and off dependent on the aging program. There are genes that are more expressed in young individuals than older, and vice versa. So the epigenome, not surprisingly, affects aging because it affects expression of genes, and we know gene expression affects the aging process. So Sinclair, when he made this original finding as a postdoc.
Let me just interrupt you for 1 second, because I think this is such an important thing that sometimes it's helpful to get a different perspective on the way I think about the epigenome. Just in a very, I always really simplify things in my head because I feel like I'm an ex-surgeon and we're not generally that smart. And tell me if this is a decent way to think about it. I think of like the DNA code itself is the letters that spell out words in a sentence or in a paragraph. Right. The epigenetics I think of as the spaces and the punctuation in between that by themselves necessarily don't do anything, but they tell you what to do with the words. So in other words, they're the less important things that we don't really think about. We just take them for granted. They're just there. But in reality, the epigenome is very important because even though it's not coding for the genes themselves, it is telling those genes how to be expressed. And if we think about it in terms of language, we will express things differently based on punctuation. So I think that I don't know if that makes it more complicated or less, but it's part of the genome. It's still a bunch of letters, A, C, T, G, and all that kind of stuff, but it's actually not coding for specific proteins. But it tells these genes, it tells these chromosomes how to fold and how to have 3D structure, and that 3D structure ultimately determines which genes are expressed and which proteins are made.
Right, so the switches that turn them on and off, and it's not fully on or off, it's tweaked at various levels, what their dynamics is, are they turned on quickly or slowly, and so forth. So yeah, I think that's a reasonable way to think about it. The only hesitation I have is punctuation implies that because back in the foundations of molecular biology, it was clear that the genetic code is not punctuated.
Right.
There was a big point of that. That's
Yeah,
the only.
yeah.
It is the word, the letters in the genes don't change.
Right, exactly.
But it would be like if you had a text where there's a whole bunch of words coded that don't make sense if all the words were there, if you read them all at once. But if you turned off certain words, let's say you scratched them out, you covered them with black ink or something, and you covered up the right ones and then looked at the text, you could read a meaningful text. And that meaningful text would be the identity of a certain kind of cell, for example. And if all those genes, all those words were not blacked out, it would be nonsense. So the epigenome is an important part of blacking out the words that are nonsense for the state of the cell that needs to be there, and illuminating those words that need to be there so that the whole thing makes sense when you read it out. I guess that would be another analogy I'd make.
And I guess the problem then is if that epigenome starts to get corrupted by various types of chemical reactions called DNA methylations, right? So maybe you want to pick up from that.
Yeah, so in fact, the chromatin, the chromosome structure that you alluded to, how the DNA is wrapped up affects how a gene is turned on or not off. If a gene is wrapped up very tightly, it tends to not be on. It's like it's hidden, it's blacked out by the ink. If it's more open, if it's not as tight, then it's on. It's as if you're illuminating that word or that gene. And the modifications to DNA called DNA methylation, which is just a little extra compound to put a little extra molecule stuck onto certain parts of the genetic code, affect the higher order structure and affect which genes are going to be expressed, not on or off necessarily, but up or down. And that, what Sinclair had talked about when he first had this inspiration was he recognized this as the information theory of aging. That is to say, there was information hiding in the epigenome, or not hiding, I mean, we can actually access it now, information in the epigenome that was specific for various periods of lifespan, and that it changes and you could say breaks down, becomes less faithful, becomes less accurate, breaks down for reasons we still don't understand, I think, as a cell gets older.
So to a certain degree, the information theory also suggests then, like when you're younger and you don't have damage to the epigenome, that a liver cell is 100% liver cell, but over time there may be some corruption and different genes that come, so it becomes maybe slightly less a liver cell than it used to be, maybe have some of the components of a different type of lineage. And that sort of the corruption of the pure information.
Right, that's one, and it may not actually be switching from a liver cell to something else. It's just not as good a liver cell.
It's not as clear.
The information is not being read. It's noisier. The information has noise in it. Noise just means variation. We think of noise as being sound noise, but noise in information systems means a less faithful, less accurate, or non-information content component to the information, non-useful information. So what you see, in fact, is that the gene expression in an old liver cell is not necessarily, for a lot of genes, is not necessarily out, down, or up. It's just noisier. It's more down and more up. It's not as tightly regulated. It's not as well regulated. And in the epigenome being the same way, it's not as clear-cut as it had been in youth.
So Sinclair, I mean, I think, you know, in reading his book and just listening to him talk, I get the sense this is really the holy grail for him. This is what it's all about.
Because his whole career has been about
which is
and I will say, of course, his book, and he's a very public figure, is very much directed by his notions of what aging is about.
Of course.
So it is one part of the story for sure.
So how much of it, so let's go back to then, because I know I actually also spoke to when Cynthia
Kenyon was here.
Cynthia Kenyon was here. I asked her the same question. I'll ask you, do you have any specific overriding theory of the aging process, the way Sinclair essentially has nailed this down for himself with this information theory?
My overriding theory, it's not going to be very satisfying, is that it's enormously complex, and the summation of networks of processes. The epigenome is a very important one, but there is proteostasis, for example, the removal of defective proteins. That doesn't come through in the epigenome.
Yep. Senescence.
There is maintenance, senescence, the maintenance of telomeres, the ends of chromosomes. That doesn't come through in the epigenome. Problems breaking down in stress response pathways. For example, oxidative stress. There are what are called oxygen-free radicals that damage cellular components. That's not reflected in the epigenome. And so the unsatisfying answer for me is it is a complexity of all of those and many other components, the many aspects that you can measure in aging, that collectively results in the progressive change in our age. Now, there are some that are going to be more prominent than others, for sure. I think the epigenome is really exciting and is certainly a very prominent element of it. What it comes down to at most, and maybe this reflects my own orientation as a molecular geneticist and biochemist, is it ultimately traces back, even if it's the epigenome that is manifesting the changes, ultimately comes down to the original starting condition, the genetics, what we are as a species, our genomes.
Right.
That's what it ultimately draws down to. So if I had an overriding theme, I would say there is a program in our genome, not the epigenome, but the genome that we started with at the point of conception. That program sets the right levels of all the and regulatory systems in all the components in all our cells. And if we are going to really advance, I think, our understanding of and intervention in aging, for me, it's going at the level of the genome or the products of the genome that are expressed.
Yeah.
And that would be, for example, through pharmacology. So I'm talking either gene therapy or pharmacology.
I want to go back to this notion of information theory again. You know, one of the products of that whole theory and how to implement it has been various biological clocks.
Right.
So there is something called the Horvath clock. Maybe you can explain what that is and sort of the derivations of the Horvath clock and what the significance of that is.
So Steve Horvath discovered, maybe, I can't remember, 2011 maybe or something, that in fact he could pin certain epigenetic changes of the type we're talking about, DNA methylation states, to age. That is, I don't want to do this myself because I don't want to find out what my effective age is, but he could analyze the epigenome and correlate the age of the individual with the state of the epigenome. And so it seems to be pretty strongly predictive. It gives you a sense for how rapidly you're aging. That would be the aging clock. The clock advances, the changes in the epigenome advance. They advance at different rates for different people, of course. And also different organisms, of course.
Yeah.
And that progression we could call an aging clock is measurable. Yeah. And that was the exciting thing, is it's measurable by analyzing the epigenome. So that's how I would sum up.
Yeah. So there's a few derivations of that seem to maybe be a little bit more accurate. And I think he has a newer clock too, Coker Age. But the interesting thing to me there, and just kind of would love to get your thoughts on this, is, again, it's based in this information theory. Ultimately, what they're looking at, a large component of what they're looking at is DNA methylation of those epigenetic areas. I like to think of them as sort of fingerprints, because what they're doing is they're taking people over, a large number of people over different ages, putting them using artificial intelligence, and essentially dividing them up based on what kind of fingerprints you see at various ages. And then if you go in there and you get a blood test done, and you're 35 years old and your fingerprint looks like you're 50, then that's sort of predictive that you might be dying at an earlier
that's right. You spelled it out better than that's what I was trying to indicate. Yes. Yeah. That you can predict sort of what age what your effective age is. It's a little. So I haven't done. Have you done it?
No.
Yeah. I mean, it scares me because I don't want to find. I already see the signs. I look in the mirror and I already see the signs. So I don't want to. I don't want to know that I'm aging faster, or I would love to know I'm aging slower, but I don't know if I am.
Well, along that lines, though, a lot of people are using this who are interested in the longevity space to use various kind of interventions.
Yes. Yep.
To see if that they can reverse it. And again, going back to Sinclair and some of the others that are out there as public figures in the space who are claiming to reverse their age by a year or 2 years every year. So in effect, making themselves a year younger every year, even though they're getting a year older.
Right. You're going to reach a limit there for sure. But I think there's certainly evidence that you can set the clock back a bit based on the measure of the epigenome, based on the DNA methylation state. And the obvious things that we already know about are reflected in this aging clock measured in the epigenome. And that is that obesity, of course, drives it the wrong way.
Right.
Smoking, for sure, drives it the wrong way. Exercise drives it the right way. You can actually measure this. These are not sort of your parents or your society saying, oh, you should stay thin and exercise because, we can correlate that across a population and we know you're going to live longer. You can actually see this changing in the epigenome. So it is a very useful, that's where that tool I think is extremely useful to figure out which interventions can act to change the aging clock, which interventions that we can actually do something about can change the aging of the clock.
Now, has there, and I want to say that Horvath actually had some studies where he was showing changes with various interventions in the aging clock in animals in his lab. Is that right?
Yeah, so there's been a number of studies like this, and several labs have shown, in fact, that in animal studies you can actually do things that actually sort of reverse the epigenome state, at least in artificially rapidly aging mice, for example. So, for example, you can damage the mouse genome in a way, not that breaks genes or whatever, but in non-genome gene regions that should be harmless. But that results in sort of an artificial, more rapid aging of these animals. And then you can actually reverse the process. And you can reverse the process by fooling these cells into thinking that they are basically younger cells or even embryonic cells. And you can do that with a set of factors for which there was a Nobel Prize called Yamanaka Factors. I don't know if you had questions coming up about this, but Yamanaka won a Nobel Prize for the discovery that a set of only 4 proteins, these are called transcription factors, those are proteins that act in our nucleus to change how genes are expressed. These 4 can take cells that are not stem cells, that are not cells that are able to divide many times and produce more specialized cells. They can take differentiated cells, not stem cells, and turn them into stem cells. They sort of refresh or renew the state. In a way, it's a little reflective of what I was talking about earlier.
Sure.
When a new generation starts, there's a refreshing process. And in fact, the remarkable discoveries that have been made in the last year or so, couple of years, has been that these Yamanaka factors that are involved, that were found to take specialized cells and turn them into stem cells that can then produce new organs and so forth, also seem to refresh the epigenome. They seem to reverse this aging clock, at least by measure of these methylation patterns.
Yeah, we actually have, and I did a podcast on this, on Yamanaka Factors, and a specific brief one. So if people want to go back and listen to that, just as like 10 minutes long, just review of the Nobel Prize, the why it's significant, all that kind of thing. What role do Yamanaka factors have in our bodies in general? Are they typically expressed? So what I'm thinking about a little bit is, it gets me back to thinking about your comment about gametes, right? Like when we're passing on our DNA, obviously we're not passing on a bunch of DNA that's methylated. As the epigenetics has already been corrupted with a bunch of methylations, right? So I guess there's 2 parts to that question. Is like, one is, why is that? Why is that DNA clean when it's passed on? The second part is, does Yamanaka factors have anything to do with that, or what do they have to do normally in the body?
So in the early embryo, it's more complex than that, but certainly in the creation of stem cells, which we create as early embryos, again, these are cells that can give rise to, they can keep dividing on their own. They're not specialized, but they can give rise to various kinds of specialized cells. Embryonic stem cells are those pluripotent embryonic stem cells, are those that can give rise to all of the tissues in a person, or any kind of animal, whatever animal the stem cells are derived from. And the Yamanaka factors are expressed in those cells, as are a bunch of other factors. Yamanaka took the large set that are specific to those kinds of cells and whittled them down experimentally and found that these 4 have this effect. So they have a natural role in resetting the youthful state of cells, and so they're very important when we're embryos in establishing from an older state into a newer state during embryogenesis. So they're definitely playing a role in that for sure.
Right. So upregulating, part of some of what the labs have been doing is potentially, I guess they've been upregulating or expressing these proteins that come from these 4 genes, and showing in lab situations, and actually in animals. And I know there was I think there was an optic nerve study where there was a crushed optic nerve that regenerated.
Yes.
They're basically showing that you can potentially regenerate some of these things without the DNA damage. So the Yamanaka factors somehow, when they're bringing things back to the original blueprints, are doing so free of the damage to the epigenome.
Right. Well, they reset the epigenome. And so are you asking, sorry, how do they do that resetting?
Kind of, yeah. I guess I'm asking, what do they do? Because obviously, first of all, if we talked about, let's take the optic nerve example, right? So why would Yamanaka factors help that optic nerve to grow again? I'm just thinking about that out loud, because the Yamanaka factors in that situation are sort of activating that original blueprint of genes. Is that?
Yeah, exactly. Yes. So those factors go in, change, they turn on genes that would normally be turned only on in these earlier cells that have regenerative capacity. And in turning on the genes, they also change the structure, because they bind into regions of the genome. They also actually result in changes in the chromatin epigenome, that is the proteins around the DNA, and the DNA itself, the methylation of the DNA itself. They actually go in there physically, and they bring in enzymes, they bring in factors that do the modifications, that is, do the additions or subtractions of methylation groups and other modifications to the chromatin as well. So they attract those proteins and actually physically and chemically make those changes.
So one of the I guess the most recent study, again, I don't want to make this all about Sinclair, but he had a recent study that showed various drug cocktails could, in effect, do some of the same things that the Yamanaka factors were doing without any sort of genetic manipulation in the Petri dish.
Yes,
in
cultured cells. He hasn't shown this in animals, but I'm sure these animal trials are well underway now. And we'll hear about it, and I think it looks really exciting. This just came out last month. And they've developed a very nice, fairly quick way of scanning through cells to see if they're acting more like younger cells or older cells. And so that allows you to set up large trials of compounds and combinations of compounds. And lo and behold, after screening through large numbers, they found some that actually do this. And it fits in with my notion. I know we've talked about this offline at other times about the notion that I think the answer, and I kind of alluded to that earlier, to really extending Healthspan is going to be through cocktails. There's no, and I did allude to that, it's a complex set of mechanisms. Again, the epigenome is a big part of it, but even for the epigenome, they required a cocktail set of compounds that do this. Again, we don't know if this is going to work in a whole animal. I feel optimistic. I think they may. And that's, boy, those would be the first, of course, there are many steps between making that discovery in the lab to something we can take, but those would be the first drugs that could make us younger just by popping a pill.
And I do want to emphasize that I thought when I read the paper, the first thing I thought was, Joel talked about this 6-9 months ago.
Exactly.
And said he thinks that the answer is somewhere in the cocktails.
It is.
So when I read that study, I was halfway I was like, cool, look at this, Joel. But then I was also like, damn, Joel, that was what you were thinking too.
Yeah, and that reflects the fact, of what we know, that there are lots of tweaks that change longevity in model organisms. And what we believe is that those tweaks are going to act synergistically. So that is, if you change one gene, you may not have a strong effect on longevity, on health span. You change another gene, you may not. You make both of those together, and I'm talking about gene changes now, you get a very strong synergistic effect. This is a phenomenon I've observed throughout my entire scientific career. We've seen it over and over. And drugs are the same thing. Drugs aren't mutations, they're not changes in DNA, but they can have the same sort of effect. They don't change the coding of the DNA. They just change the products that are made by the DNA, change how active they are. That is to say, the proteins, the enzymes that do all the various activities inside a cell. And again, it's a very complex process, aging is, but I think that there will be key nodes, key points that you can intervene in that collectively, and we don't know how many that'll be, act very synergistically. You know, if it's a handful, then we can actually tackle it. If it's 6 drugs, for example, and we can extend our lifespan 15 years, you know, that may happen, that may work.
Yeah, and I think that one of the key parts of this research might actually be, again, bringing that spotlight to the cocktail thing, because, you know, I've talked about on this show about a program called the Interventions Testing Program, which really looks at individual drugs to see if they have any effect on longevity in rodents, right? And, you know, some of the drugs that in the lab seem to have some effects have actually not shown significant benefit in that model, including, say, metformin, for example. Resveratrol is another one that I know that a number of longevity people talk about, did not show benefit. But what they haven't done in these protocols is to sort of, as you mentioned, look at things, could they potentially be synergistic? If you've got all these different pathways and you have something that's affecting cellular senescence on the one hand, you're doing something on the other side that's helping with, you know, the epigenome, that putting those together is amplifying that benefit. And
that's what I meant by synergy, actually, synergistic, that if each one has an effect of 2, and you use both of them and you get effect of 20, that's we see that over and over and over in cellular processes, for sure. And the problem with drugs is that my understanding, now I'm not in biotechnology, but my understanding, or pharmaceutics, but my understanding is the FDA is that developing clinical trials with single drugs there's a pipeline for that. For cocktails is much trickier. That is, unless they're already, I think if they're already FDA approved, it might not be such a problem. But if you have 2 completely new drugs, getting a clinical trial approved for combinations is really tricky. That's my understanding. And so one way to fool the system then is to make a single drug out of 2. So for example, these compounds that Sinclair's lab has discovered, if they were all hooked up into a single molecule, that would be considered a single drug. So it's kind of getting around the bureaucracy in a way.
That's right. That's right.
Okay, we're going to stop right there for now. Not everyone has an hour and a half to listen to a single show, so we'll have the rest of the interview on the next episode of Sapio with Buck Joffrey.